you. Sorry, microphone issues. And I have a lot of things up on my computer that luckily aren't showing. So first, thank you to Esper for inviting me to this beautiful, beautiful country. I have absolutely fallen in love with Brazil, and this has been a wonderful meeting, so thank you. So I am, hopefully, this works. Sorry, this isn't changing. The slides won't change. My apologies. These won't change either by this or this. It's dinging at me. Let me try this one. Is it going? There we go. Okay, wonderful. So I'm going to talk to you today about the microbiome. And the microbiome is in several areas of the body. So you have microbiomes in basically every mucosal tissue, so things like the mouth, the nasopharynx. The gut is the largest microbiome, so there's actually 5 to 10 pounds of bacteria in your GI tract. The reproductive tract is a major site for the microbiome, including the female reproductive tract, the vagina, cervix, and ovaries have bacteria in it. A distinct microbiome in the foreskin and penile and urethra. Potentially in the placenta, there's some controversial data on this right now. But even your skin has a microbiome, your ears, and your lung. So just to take a step back and talk about what the microbiome is, just wanted to go through some definitions to make sure we're all on the same page. So microbiota is the communities of the microorganisms. So this comprises all of the different microorganisms, including bacteria, archaea, eukaryotes, so this is like fungus and protists, and viruses as well. But then when we talk about the microbiome, this is the entirety of microorganisms, including not just the microbiota themselves, but also their genes and functional products, which we'll talk a bit more about later. And then I'm going to use the word a lot today of dysbiosis. So dysbiosis is when you have an imbalance of the, I say intestinal here, but any of the microbiome. We'll also talk about dysbiosis in the vagina later as well. So when you have qualitative or quantitative changes in your microbiota or the products made by your microbiota, this is when we have dysbiosis, which is a common feature in HIV, and I'll discuss more for both pathogenesis and transmission. And this doesn't want to switch again. There we go. So just to briefly orient you to microbiome plots, as I'll be showing a lot of them, particularly these abundance plots of diversity. So when I show you a plot like this, each color represents a distinct different bacteria, whether it's a species or genre, but each color is very distinct. And then the overall diversity index is typically below, showing the higher this bar is, the more diverse, as you can see. So we have a lot of colors here. That's going to be a very diverse microbiota, whereas there's not as much color here. That's going to be a very non-diverse microbiota. Another thing we like to talk about a lot with microbiome is principal component analysis, or it's also called PC or PCA analysis. And this is just showing how distinct each community is and whether they cluster together. We like to say they cluster because there's similarities. So here I'm just showing you an example where we have a jejunum in blue, colon in these red triangles, or rectum in orange, and you can see that the jejunum is more distinct as it clusters more over here, whereas the colon and the rectum cluster more together over here. So, and this is not any data, primary data that you need to worry about, just trying to orient you to how to look at these plots. So what happens with the microbiome in HIV infection? So this is the GI microbiome from uninfected or HIV-infected subjects. And now I'm showing you another relative abundance plot. So instead of the multiple bars I showed earlier, this is just one bar averaging multiple patients. So what you can see if you just look at these bars is there's of course differences just seeing them, but when you actually quantify them, there are significant changes in a few things, including increased prevotella in HIV-infected individuals and decreased bacteroides. I will talk about more what that means, but the point is that when you look at HIV-infected subjects, there are key statistically significant changes in the microbiome. So this is a cartoon of how these things are interacting, what's going on in the GI tract during HIV pathogenesis. So in a healthy GI tract, you have an intact tight epithelial barrier, and you have a healthy microbiome that has lots of good bugs like firmicutes, lactobacillus, different bugs that we know prefer healthy microbiota that are important for homeostasis. And then you have homeostatic cells and cytokines, such as Th17 cells. And this is really important for the balance and maintaining the integrity of the GI tract. In the context of HIV pathogenesis, however, you have damage to this tight epithelial barrier, and this is associated with multiple dysfunctions, including increased pro-inflammatory cells and cytokines. We're going to talk a lot about neutrophils in a moment, and then this altered microbiome. Later on in my talk, I'm going to talk about transmission, but I'd just like to show these back to back, as there's very similar mechanisms that underlie both HIV pathogenesis and mechanisms that lead to increased HIV transmission when we look in the female reproductive tract. But I'll get back more into transmission towards the end of the talk. So like I said, there's these overlapping mechanisms that are associated with both transmission and pathogenesis, and this includes the barrier damage, the tight epithelial barrier I showed, inflammation, and an altered microbiome. So we're going to start with the pathogenesis. So I think this was very well covered in our cure talks yesterday, but why do we care, of course, about pathogenesis? And this is because HIV-infected individuals, despite suppressive antiretroviral therapy, still have increased morbidities and mortality compared to HIV-uninfected individuals. The reason I'm so focused on mucosal tissues in the GI tract is because, despite this antiretroviral therapy, what is consistently associated with increased morbidities and mortality is microbial translocation and markers of mucosal dysfunction and damage. So we've seen there's gut epithelial barrier dysfunction and innate immune activation that's associated with barrier dysfunction, and that is associated with mortality, inflammatory and coagulation biomarkers, and this includes microbial translocation markers, and again, soluble CD14, which is an innate immune activation as well as microbial translocation marker. So I think what really well shows or well demonstrates what happens in the mucosal dysfunction that happens during the HIV infection is by using the monkey model of SIV infection. So this is a healthy macaque, a healthy rhesus macaque colon. And I know it's probably hard to see with the lights. But in green, what you have is cytokeratin, which labels that you have a nice, intact, tight epithelial barrier of the colon as you'd want. The blue is just the DAPI, the cells back down here. And importantly, red is a marker for E. coli, and it's kind of a pan-gram-negative bacteria marker. And what's important is that this bacteria is in the lumen of the colon where it should be, and there's an intact, tight epithelial barrier. When you infect a rhesus macaque with SIV, however, of course, the model for HIV, what happens is where these arrows are, you can see that there's focal breaches in the cytokeratin because there's focal breaches that tight epithelial barrier. Where those breaches occur, the microbial products, again, I know it's hard to see, but there's red coming through into the lamina propria. So these microbial products are actually coming through that tight epithelial barrier. In addition, you can sort of see that there's more of the bacterial products. So we have more E. coli, more gram-negative bacteria, which are not good bacteria. I'm sorry, I'm going to try to keep having warnings coming up on my computer, which I believe is why this keeps messing with the presentation. So one of the things that we've worked really hard on is trying to understand the mechanisms of mucosal dysfunction in HIV infection. And what this has led us to is these studies of the microbiome. So during HIV infection, you have this microbiome dysbiosis. So in a nutshell, what I want you to remember that we're looking at here is that in HIV infection, you have increased these bad bacteria. So increased Prevotella and increased proteobacteria. So Prevotella are bad because they're highly inflammatory. They induce a very significant immune response. And then the proteobacteria are a lot of our opportunistic pathogens. So things like Salmonella, E. coli, things that are associated with disease. And another important point about proteobacteria is that these tend to be very adherent bacteria, so they can actually latch on to that GI tract, which allows them better to break down that epithelial barrier and get through. So while we have this increase in these bad bugs, we also have a decrease in good bugs. So we lose our firmicutes and our bacteroides. So firmicutes are very important. These are the bugs that tend to be probiotics. So things like Lactobacillus that we know are very healthy and maintain a healthy epithelial barrier and are constantly associated with good responses. The bacteroides then is another class of bacteria that make a lot of things called short-chain fatty acids that are very important for maintaining mucosal immunity in the GI tract. So we lose these good bugs, we increase our bad bugs, and this has been associated with pathogenesis. But I want to point out something very important here, and that is that there's a recent study by Roger Paredes showing that when you look at HIV-infected individuals that are men who sleep with men, compared to HIV-infected individuals who are heterosexual, you see more dysbiosis. And so then he looked at uninfected MSMs, or men who sleep with men, and found that they still see dysbiosis. So there is a chance that the dysbiosis I'm describing to you in the during HIV infection might actually have been pre-existing before infection when we look at men who sleep with men, MSMs. But I still think it's really important to study this because I still think that regardless if this happened before infection or due to infection, there's still impacts on infection, and we see this, the dysbiosis is increased in HIV, so it's very important. So just to go back to this dysbiosis I'm telling you about and really drive this home. So now we have an HIV negative on this side. So again, healthy microbiome. We have things like butyrate, vitamin K and B, these very important factors that are made by our healthy microbiome, like our Bacteroides and our firmicutes. We have protective IgA here. We have an intact barrier and healthy cells. When it comes to HIV infection, we have decreased barrier proteins, decreased protective IgA, and then we have this loss of our good bugs and an increase in bad bugs like proteobacteria and Prevotella. This also leads to increased things like LPS and hydrogen sulfate production, as well as endotoxins, which are constantly associated with increased microbial translocation and increased morbidities and mortality. We also have this increase in activated leukocytes, and particularly neutrophils, which I'm going to get into more now. So there's this very important relationship between the cells and the microbiome, and there's a lot of crosstalk here. And particularly neutrophils are important because neutrophils are the most abundant immune cell, and they are very fast responders to bacteria in particular. So they can respond to virus as well, but they are classically known as bacteria eaters, and they're the first cell on the scene. So we study neutrophils because they're so interactive with bacteria, but also because they are so abundant, and they are extremely understudied because they're very difficult to study. You have to do everything prospectively, as they don't survive freeze-thaw. So neutrophils have 3 ways that we know of that they are antibacterial. They can phagocytose the bacteria directly, they can degranulate and trigger death, or they can make these nets or neutrophil extracellular traps. What's important to remember about neutrophils is that while these antimicrobial functions are really important, it's always a scale because neutrophils are also highly inflammatory. So neutrophils release lots of pro-inflammatory cytokines and lots of proteases, and the whole function of a protease is actually to break down barriers. So if you have neutrophils that hang around too long, you end up with tissue damage and other exhaustion and suppression of cells around you. So we need to make sure these neutrophils are gone right away. And this is why, if you know about neutrophils' half-life, they die very quickly. They respond and die. So they're the first ones in, first ones out, so you don't end up with tissue damage and T-cell suppression. However, in HIV infection, it's been shown that you have accumulation of neutrophils during HIV infection in the GI tract. So the brown here is myeloperoxidase, which is a marker for neutrophils, and you can see that there is an excessive amount of myeloperoxidase in HIV-infected colon compared to HIV-negative. And this is just quantifying this, and you can see that whether it's a viremic HIV-infected person or someone on ART, whether immunological responder or non-responder, they have significantly more neutrophils than someone who's uninfected. We also know then that there's this damage to the barriers I showed earlier. And we can see in HIV uninfected individuals, you have this intact barrier, and in the context of HIV infection, again, you get these focal breaches in the tight epithelial barrier. So we thought maybe there's an interaction between the increased neutrophils that we know can cause tissue damage and then this damage to the tight epithelial barrier. So we sought to specifically address how many neutrophils are there if we quantify them by flow cytometry, which is going to be more specific than just looking at myeloperoxidase. And then we wanted to understand what they're doing and why they're there. So when we measure neutrophils by flow cytometry in the GI tract of HIV-infected, and these are all ART-suppressed individuals, you can see that there's a significant increase in neutrophils in the GI tract. What's important is, like I said, a neutrophil has to die very fast in order to clear out and not cause tissue damage. So we looked at active caspase 3, which is a marker for apoptosis, or death of the neutrophil. And what you can see is that while you have a lot of apoptosis in an HIV-uninfected person, this is significantly reduced in HIV-infected people. And importantly, there's a significant negative correlation between how much caspase-3 is expressed and the total number of neutrophils in the GI tract. So this indicates that there's a relationship, as would be expected, between the death of these cells and accumulation of these cells. So we wanted to understand what mechanisms underlie the neutrophil accumulation and decrease apoptosis during HIV infection. To do this, we took whole blood or isolated neutrophils, and we looked at how bacteria that we know are altered in HIV infection that I showed you earlier might be affecting the neutrophils. So we looked at 3 bugs that are increased in HIV, 2 Prevatellas and a Cinetobacter, and 4 that are decreased in HIV, Ruminococcus, Lactobacillus, and Bacteroides. And then we did a lot of controls for different TLR stimulations to try to understand really what's going on here. Once we co-cultured the bacteria with the blood, we then assessed the neutrophils via flow cytometry. So I know this is a very busy plot, so to summarize, we have no incubation, and then this is no stimulation, so this is just media. So this is going to be our best example right here that we're really comparing, and all the statistics are compared back to this. So the no stimulation is just if you have a neutrophil at steady state. We looked at how HIV itself, we used inactivated HIV to see how HIV itself might affect the neutrophils. Wasn't a huge difference. Sorry, and this is looking at apoptosis of the neutrophils because we're trying to understand why they're dying. But then when we look at gram-negative bugs that are increased in HIV infection, you see that we start having decreased apoptosis, whereas when we have the lactobacillus up here, so the good bugs that are lost in HIV infection, you have a normal level of apoptosis or even higher apoptosis. So this is really important because it shows that we can actually directly drive apoptosis by good bugs, so we want the apoptosis, we want these cells to die, but that the bad bugs that are increased in HIV infection might actually be delaying apoptosis or decreasing that apoptosis. I told you that we did TLR stimulation, so we really wanted to understand what TLRs might affect this. And when we look at TLR4 and TLR2, this is how we see that we can decrease this apoptosis. So we believe that this dysbiosis occurs, and then it's driving a neutrophil response because you have these bad bugs that can get into the barrier that's breached, and now we have delayed apoptosis, probably in a TLR-dependent manner. So to conclude this part of the talk, neutrophils are increased in the GI in HIV infection, and this might be also driving the tissue damage that's there. And homeostatic apoptosis of the neutrophils is decreased in the GI tract, likely due to interactions with the bacteria, such as Prevotella and other gram-negative bacteria that are increased in HIV infection. And this might be the mechanism underlying increased neutrophil accumulation in the GI tract during HIV. So now back to our overlapping mechanisms that I talked about in transmission and pathogenesis. So I showed you how barrier damage and an altered microbiome can potentially drive inflammation, but now I want to go back into transmission and talk about some of these mechanisms as well. So it's really important to remember when we think about transmission that women are a highly vulnerable population, particularly in sub-Saharan Africa, that are getting infected with HIV. Women tend to get infected at a younger age than men. And there's 2 times women ages 15 to 24, so very young ages than men, that are getting HIV infected. Women are highly vulnerable due to things like sexual violence and lack of condom or sexual protection negotiation rates in Africa. And then this is also important to think about women because it's not just that they're getting infected, but there's also mother-to-child transmission. So back to my cartoon of these interactions. So now I'm showing you different mechanisms that have been associated with increased HIV transmission. So this is not pathogenesis now, this is just transmission. So in a healthy female reproductive tract, so in the vagina, you have the squamous epithelial barrier, so it's a lot thicker than in the GI tract. And of course, it should be intact to be protective. In a healthy vagina, you have predominantly lactobacillus. So lactobacillus is extremely good for the vagina, and if you have a domination of the lactobacillus, it's associated with better health outcomes. And then again, just like GI tract, you have homeostatic cells and cytokines that help maintain this barrier. Different mechanisms that have been associated with HIV transmission include then this altered microbiome, which I'll talk more about. Again, these breaches in the tight epithelial barrier, increased pro-inflammatory cells and cytokines, and again, you have neutrophils at play, but we're not going to talk about that for the rest of this. So what is the clear underlying factor, though, of transmission is that inflammation is a key factor, and this has been shown time again, that the higher genital inflammation there is, the increased risk of transmission. One of these underlying causes of inflammation could be bacterial vaginosis. This is also known as BV. So BV is the most common cause of vaginitis. It's characterized by loss of the Lactobacillus species that I told you are so important, with increased diversity of different vaginal microbiota. Different bugs like Gardnerella, Microbiome, multiple prevatellas, and other BV-associated bacteria that I'll get more into momentarily. What's important to remember about BV is this does not just affect women. So while a woman who has BV is at a 60% higher risk of getting HIV infection, this also affects female to male transmission, as a male that is sleeping with a woman who has BV has a 3-fold higher chance of getting HIV infected, and this also increases mother to child transmission. So this is just how we measure BV clinically. This is a Nugent score. A Nugent score of 0, we're seeing a lot of these lactobacilli, and there's no other organisms or cells. In BV, this is a high Nugent score of 10. You have these polymicrobial preparations, so lots of different bugs. There's these specific cells called clue cells, and we lose our lactobacillus. To show this on a very busy RDP plot now, so this is our diversity. So remember I told you one color means low diversity. Orange, yellow, and red are lactobacillus. So if you have lactobacillus and you're BV negative in green, you have this predominant of 1 or even 2 bugs, but there's very rarely anything but lactobacillus. However, in the context of BV, or vaginal dysbiosis, you have multiple species, and this is what I was telling you about. There's things like Gardnerella, prevalence. So I'm going to talk a lot about Gardnerella from here on out because Gardnerella is almost always associated with BV. So one of the things that we wanted to understand is how these bugs might be affecting the epithelial barrier. So we did a wound healing assay where you scratch, these are HeLa cells, so cervical cells in culture, and we grow them to confluence and scratch it to induce this wound. And you can see that with no bacteria, it's going to heal well in 24 hours. What's interesting, so if you put lactobacillus, these are the good bugs in, it will still heal, but if you put Gardnerella vaginalis, so this is actually just a super name from the bug, but it happens when you put the live bacteria in as well, you don't get healing. So this is one of the first things we found, and was recently published, that if the bacteria can directly affect that epithelial barrier and the ability for it to heal and stay intact. But we wanted to understand more how this Gardnerella vaginalis, or the vaginal dysbiosis in the female reproductive tract, was actually affecting HIV. Because what's important is that the areas where women are at highest risk for HIV in sub-Saharan Africa, you have a lot more bacterial vaginosis than you do in areas where women are lower risk, like North America. So we have our Gardnerella, or BV, is in this purple-blue, and then our orange and yellow are lactobacillus. So in North America, the women are almost predominantly lactobacillus, with much less amounts of women that have this BV or the Gardnerella dominant. Whereas you can see that the orange pie slices are much smaller when we look at, and these are from different cohorts in Africa that it's been measured. So there's a lot more abundance of this Gardnerella, this BV phenomena in Africa compared to the US. So we want to understand how this diversity in the vaginal microbiome in areas where women are high risk might affect their protection efficacy. And that's because there's been a lot of protection trials using PrEP, or pre-exposure prophylaxis, in women, and there's highly variable results. So while PrEP tends to work very well in MSM, in men, to prevent HIV infection, it's extremely variable. So effectiveness is 0 here, and these are different trials that have been done to look at PrEP. And you can see that in a few of the trials, and these are actually in partners, so these are in discordant people who are in a monogamous relationship, so they're going to have the highest protection anyway. So they're up to about 80%, but then these other studies that aren't in discordant couples have anywhere from negative 60% to 60%, so it's very variable. And I'm going to talk mostly about the Caprisa 004 trial here. So this was a coital tenofovir gel. So women were given a tenofovir gel, and tenofovir is an antiretroviral drug, and were asked to use it pre or post-sex within 6 hours. And they had a very low, so about 40 to 50% efficacy in this trial. So just go over the trial design of the samples we looked at. We had 889 that were analyzed in this trial, and several were excluded either because they didn't give consent for samples or other issues. And how this comes out is that we ended up with 688 participants that were analyzed, and these were equally in tenofovir gel group or the placebo gel group. We then split them, and I'll show you how, into lactobacillus or non-lactobacillus, and then looked at how many acquired HIV infection in each group. So we measured the microbiome by proteomics here. So we used actually a mass spectrometry, not a 16S approach, which it does overlap very well. But again, you see that this very low diversity lactobacillus dominant, and it comprised most of the women. And again, this was over 50% lactobacillus. We call her lactobacillus dominant. Then we have this more BV, highly diverse, and the purple is Gardnerella, and you can see some very Gardnerella vaginalis dominated here. And we grouped these women into this less than 50% group. So these were non-lactobacillus dominant. So NLD is non-lactobacillus dominant, LD is lactobacillus dominant. We then looked back at, we wanted to split these women into the groups and now look at the efficacy. So not just the overall efficacy that was done for all the women in the trial originally. When you split women into group, what is really fascinating is that women who are lactobacillus dominant, so this is only lactobacillus dominant women here, you see that there is a significant efficacy of the tenofovir gel compared to the placebo. However, when you look at the women that are not lactobacillus dominant, you lose that efficacy. So while this trial was originally, when you look at all women together, they said it's not an efficacious trial, this did not work, what we found is that actually if you look by the microbiome, if you have a healthy microbiome, the tenofovir gel, it did in fact work, with 61% protection in women with lactobacillus and only 18% protection in women who did not have lactobacillus dominant. So what was very interesting then is we went back to look at what was happening. So a lot of these trials were blamed on adherence. So many of these trials said, well, they weren't efficacious because women aren't being adherent with their tenofovir gel or whatever PrEP mechanism they're taking. So we went back to look, we ruled everyone out that were reported not adherent, but then we went and looked at how much tenofovir gel was actually in the vagina of women who are lactobacillus dominant versus non-lactobacillus dominant. And what was amazing is that if you were not lactobacillus dominant, you had far less detectable tenofovir than women who are lactobacillus dominant. So we thought, well, this is weird because I don't think that women are going to lie about their adherence just because they have a different vaginal microbiome. So we started thinking about what might be happening here and why you would have differences in your tenofovir levels in the vagina based on what your microbiome is. So we did a previous study where we actually looked at the functionality of the microbiome in women who are either lactobacillus dominant or not lactobacillus dominant, and this was in a different cohort from Kenya. But one of the things we found is that when you look at functional changes, there's multiple bacteria that are actually associated with drug metabolism that overlap cells that are associated with drug metabolism. And what's very interesting about this is that when we looked at tenofovir is actually a purine analog. And a lot of the proteins that we found in the bacteria, the functional proteins, were capable of degrading purines. So we hypothesized that there might be actually a direct interaction with the bacteria and the drugs due to metabolism. So we did an in vitro culture assay to determine if tenofovir could be metabolized by the bugs. And so we did either lactobacillus or Gardnerella, plus or minus tenofovir, and we also had abiotic controls, which was just the same media with the tenofovir. We cultured these together, and I'm going to show you later too, we did a lot of other bugs as well, but we cultured them together and then looked at tenofovir levels either in the supernatant or the cells and what potential metabolites were there. So just to orient you quickly, abiotics going to always be in the black line. Our good bugs are lactobacillus. We have both inners and crispatus are in our blue lines, and the Gardnerella vaginalis is red. And what you can see is that when we culture the Gardnerella in the presence of tenofovir, the tenofovir levels quickly go down in the supernatant. So this is only at 4 hours, how rapidly this goes down, where you have less than 50% of your tenofovir left after 24 hours in the presence of Gardnerella, but not with lactobacillus or not when it's on its own. Concurrently, you have this increase in cells. So these are inside the bacteria cell. So the bacteria cell seems to be uptaking tenofovir, where the Gardnerella can, where the lactobacillus does not. So we thought, okay, well, is there an interaction here, or the bacteria actually metabolizing the tenofovir? So we measured putative metabolites that were predicted by a tool called ChemTool. And one of the things that we saw is a predicted metabolite of tenofovir metabolism, if you do purine metabolism, results in the breakdown to adenine with the release of the oxymethylphosphonic acid. So when we measure this, we find that in the same ratio as we lose tenofovir, we gain adenine in the presence of Gardnerella, but not in the presence of lactobacillus or abiotic controls. And importantly, when we measure tenofovir plus adenine, we can see in all all cultures that we get the full drug recovery. So we know that the adenine is the major metabolite of tenofovir metabolism. And these data show that the bacteria can actually directly metabolize our antiretroviral drug tenofovir. So we also wanted to see, since we were just using a couple bugs for that, we wanted to see other bacteria that are associated with HIV infection and bacterial vaginosis. So we measured multiple prevatellas, ammobiluncus, and E. coli. And again, we see that these are all capable of metabolizing tenofovir. So all of these mostly anaerobic bugs that are associated with bacterial vaginosis are capable of metabolizing the tenofovir. So finally, we wanted to understand what the kinetics of this tenofovir biodegradation were. So what's important is tenofovir is not actually the pharmacologically active form of the antiretroviral. So it actually gets uptaken into the target cell, and then it becomes tenofovir diphosphate. So we call it the TFV-DP. This is our pharmacologically active form of tenofovir. So we wanted to add target cells into the equation here to try to determine how, if the bacteria can metabolize the tenofovir before the target cells can actually take that up and make it pharmacologically active. So we add the tenofovir with Jurkat cells as our targets, and then either Gardnerella or Lactobacillus, and then we're measuring the supernatant tenofovir, intracellular tenofovir diphosphate, so this would be into the Jurkats where it's pharmacologically active. Intracellular adenine, so this is interbacterial adenine, so if the adenine is present, that means it's metabolized the bacteria, and then, again, the total drug recovery. So, again, we have our black is our tenofovir alone, so kind of our abiotic, and then the blue here is our lactobacillus, or our Jurkat cells alone in the tenofovir, and you can see, as you'd expect, the tenofovir is decreasing because those target cells are taking them up, taking the tenofovir up. But what's interesting is you have a more rapid decrease and a more pronounced decrease in the presence of Gardnerella. And when we look at the adenine is formed in the presence of Gardnerella, but none of these other bugs or in the abiotic controls. So adenine is only formed if Gardnerella is there. So most importantly, when we look at the tenofovir diphosphate formation, so this is, again, pharmacologically active, you have an increase in the diphosphate formation with the lactobacillus or the abiotic, but not as striking with the Gardnerella. And then when we look at our total drug recovery, we can see that we get the full drug recovery. So this means that the Gardnerella can actually metabolize the tenofovir into the adenine before the Jurkat cells can actually take it up and make it diphosphate. So again, we believe that this is one of the mechanisms underlying this decreased tenofovir efficacy. So finally, just to show you how rapid this is. So this is a timeline with 15 minutes, 30, and 1 hour. Again, black is the abiotic, the tenofovir plus the Jurkat cells. Red is the Gardnerella. And what you can see is this is just looking at the pharmacologically active tenofovir diphosphate in culture. And you can see that this is significantly lower. As all of these are increasing over time, the Gardnerella can maintain significantly lower and very rapidly. So finally, we identified here a microbial signature associated with high effectiveness of vaginal PrEP, and that was that if you have a lactobacillus, a healthy vaginal microbiome, you have 3-fold higher gel efficacy, whereas it was not affected in women that did not have lactobacillus communities. We found a putative mechanism for this is that tenofovir is rapidly depleted by Gardnerella as well as other BV-associated bacteria, but not by lactobacillus, and that the active tenofovir is decreased in the presence of Gardnerella, which is much more rapidly than the uptake by the target cells occurs. So this just highlights the importance of both adherence and vaginal health for PrEP efficacy. So finally, just to thank my lab and my close collaborator, Adam Bergener, who ran all the proteomics for the CAPRISA study, CAPRISA and all our clinical partners. And with that, I'd be happy to take any questions. Obrigada.